Acetylcholine and noradrenaline are the most common neurotransmitters in humans. Because the receptors for the neurotransmitters are located on the postsynaptic membrane, the direction of the nerve impulse transmission is maintained. Binding of the neurotransmitters to the receptors generates a nerve impulse on the postsynaptic membrane. This impulse can then travel along the membrane of the postsynaptic neuron. The generation of a nerve impulse in the postsynaptic membrane is possible because the receptors located there are connected with ion channels. Binding of the neurotransmitter to the receptors causes these channels to open, resulting in a flow of ions and the generation of an active potential. Note that the synapse is where electrical transmission of the nerve impulse is converted into chemical transmission and back again. The neurotransmitter is either broken down or taken up again by the presynaptic neuron. Unlike other cell junctions, the synapse is a site where the nerve impulse may be modified. Firstly, the amount of neurotransmitter in the synapse may be regulated by the frequency of the nerve impulses reaching the presynaptic membrane and the metabolic activity of the presynaptic neuron producing the neurotransmitter. Secondly, the neurotransmitter itself can regulate its amount in the synaptic cleft. It does this by binding to receptors located on the presynaptic membrane, which inhibits its further secretion to the cleft. Thirdly, additional substances secreted to the synapses, known as neuromodulators, can regulate the frequency of the impulse generated by the neurotransmitter in the postsynaptic membrane. Finally, the neurotransmitters can enhance or inhibit the conduction of an impulse across the synapse. Because they present so many opportunities to modify neurotransmitters, synapses have become the target of a large number of drugs. These drugs modify not only the action of nervous system, but also the organs innervated by neurons in which these cells form synapses.